Very good morning, students. So today we are starting with one of another very important topic in the circulatory systems, that is the neurogenic and myogenic hearts. So students, in this particular lecture, we'll be talking about these two types of the hearts, which are based upon the innervations, or you can say the pacemaking systems of the heart. So if I take the very first case, this is a neurogenic kind of a heart. This particular heart will be having the innervation of the nerves. So here we'll be talking about that how these hearts they are pacemaking, what is the pacemaking system, how they are beating. So if I take a heart into account, we had talked about the evolution of the heart. We have talked about the evolution of the aortic arches. Now, these are the types of the heart which are based upon the mechanisms that how they are making their beating system. That if a heart, it is requiring a wave, or you can say a depolarization wave to contract or to work. Now, the question is that how that particular wave is formed in the heart. What is the seat of the formation of that wave? Who is making that wave? So, in that particular aspect, on that basis, we have got two types of the hearts, which are the neurogenic and the myogenic hearts. So, before starting this particular topic, I must give you an overall idea that in the case of the humans, we have got the myogenic hearts. But if you take the case of the invertebrates, so there we have the neurogenic kind of hearts. Now, straightforwardly, now we are starting with this particular topic. Now, if I take the very first case, that what is a neurogenic heart? Now, in this particular case, the word neuro refers to the nerve cells. So there are present a number of nerve cells. And genic, it refers to the generation of the impulse. So if the generation of the impulse or the beat, it originates in the ganglionic nerve cells, then it is a neurogenic kind of a heart. So here I have written as the ganglionic nerve cells. So if the beat of the heart is originating in these nerve cells, it is a kind of neurogenic heart. Now, this is a very, very important thing to note, that the ganglionic nerve cells, which are present around these hearts, they are an extracardiac nervous tissue. Now, here I would like to emphasize on that what is extracardiac nervous tissue. Now, students, our heart is made up of muscular tissue, that is a special cardiac tissue which is having high vascularization and high blood supply, and every heart it needs some you can say the blood supply for its own working. So the tissue of the heart, it is basically the muscular tissue. We don't have a nervous kind of a tissue in the heart, but we have some nerves which are innervating the heart walls, as we have the SA node, the AV node, or the bundle of His. Then we have the Purkinje fibers which are present in our heart. They are nervous tissue kind of a thing which is present in our hearts. But here, the ganglionic nerve cells which are making the impulse for the neurogenic hearts, they are an extracardiac nervous tissue. That means they are not present inside the heart. They are present towards the outside. They are outside the heart, and they are not a part of the muscular tissue of the heart as it is seen in our case. So I think you are clear about this. So they are the extracardiac nervous tissues which are forming the beat for this kind of heart. Now here, the wave, it spreads over the heart. Now this is again a very important part. The neurogenic hearts, basically they are not the hearts like we have. These are mostly the tubular hearts. If I take the case of suppose arthropods, or if I take the example of the cockroaches, they have got neurogenic hearts. Now the neurogenic hearts, they are mostly long and tubular kind of hearts. And if they are long, so they should have some extra cardiac nervous tissue which is controlling the contractions of that particular heart. Now suppose if there is a tube-like heart and it has to contract from the hind end towards the anterior end, and this heart is pumping the blood towards the head side, suppose. So it has to make a wave kind of contraction so that the posterior side of the heart, it contracts first, and then the wave spreads to the whole of the heart towards the anterior side. So that's how the control of the beat of the heart, it is under the extra cardiac nervous tissue which is present over that heart. And whenever this nervous tissue, it is stimulated by some impulses which are made by this extra cardiac tissue, the heart at that particular level, it will contract, and that would result in a wave of contractions in that kind of a tubular heart. So in the coming slides, we'll be seeing that how the wave it is spreading on the heart. And why I'm taking this particular point is that because these hearts are quite long in the bodies of the invertebrates. So that's why a wave is needed to to make a particular directional movement of the blood. Okay, students, if we go further, the crustaceans, so we had talked about arthropods, but this is a particular group in the arthropods. The crustaceans, they are known for their neurogenic hearts. So the seat of the origin of the heartbeat, it is in the ganglionic nerve cells, which are not present in the heart but outside the heart. Now students, if I take a particular case of animal, so I am taking here the example of limulus, the king crab. So it has got a neurogenic heart. So I have written a short form as NGH. So this is a neurogenic heart of limulus. Now here students, you can very well see that this is the heart of the limulus, which is present in between the diagram. So this is the heart. This is the aorta, first of all, where the blood will go like this into this direction. And this is the heart. This is a tube-like heart. This is quite a long heart. And these are the ostia, which is present in each and every segment, you can say, of the heart. So here the heart is long, tubular, you can say somewhat chambered, and it has got many ostia. And from these ostia, the blood can go towards the inside of the heart. So it can go from the ostia into the heart. So that's how the blood is filled, or here I should say the hemolymph is filled into the heart. Now this heart, it starts pumping from this particular site, and a wave of contraction, it goes like this. The first it contracts from here, then from here, then here, and the wave of the contraction, it goes towards the anterior side, and whole of the blood is pumped to the head side. So this is the type of the contraction which is present in the limulus. And here we have some ganglionic nerve cells. So here we have written as, like you have 7 and 8, 9, 10, 11, 12, and 13. So there are 13 pairs of the ganglionic nerve cells which are present in the heart of the limulus. Now, if I go further, the nerves, they are innervating the heart for stimulation at different parts. So here you can very well see the nerves from 7th and 8th ganglion or ganglia, they are going for this side of the heart and they are penetrating this side of the heart. And the 1 to 6 ganglia, they are not present over here. If I go for further in this particular heart, the ganglionic nerve cell number 9 is penetrating with its nerves onto this particular site. So what will happen? That if you have suppose 1, 2, 3, 4, 5 and 6 over here, first of all the impulse will travel into the ganglion number 1. It will give its nerves to this part. Now this part of the heart will contract. When the impulse it reaches 7 and 8 ganglionic nerve cells, this part of the heart will contract. When it will flow to the 9th ganglion, this part of the heart will contract. That means that will be a wave of contraction and the blood will be flowing towards the head. So that is the kind of, you can say, contraction in these types of neurogenic hearts. So here, these ganglionic nerve cells, they are not present inside the heart. They are an extra cardiac tissue. So they are present outside the heart. Now, if on the contrary side, on the other side, if I take the myogenic heart into account, so what we have is that we have a typical heart like this. So that is a mammalian heart. You can draw a mammalian heart. And here we have got 4 chambers. So these are not tube-like, first of all, I should say. Then if I take the case of the pacemaking system, so what we have is that a cardiac muscle is modified to produce the excitatory wave. Now this is a very, very important point. So students, what happens that when the heart is developed in an embryo, first of all, it is a muscular organ. So all the tissue which is present in the heart, it is muscular in the type. So what happens after some time, that some of the cardiac muscles in the heart, they are modified into the nervous tissue. And that's how we have the different types of the pacemaker units in our hearts developed in the embryo. So here I have written that a cardiac muscle, it is modified to produce the excitatory wave in the case of myogenic hearts. So I should say that these, if we talk about the first, you can say, the part of the pacemaking system, which is the SA node or the sinoatrial node, it is not an extra cardiac tissue, like in the case of the neurogenic hearts. So those ganglionic nerve cells, those were present outside the heart. But here we have the SA node very particularly present in the wall of the right atrium. So it is not an extra cardiac tissue. Now, how it is formed? It is a modified cardiac muscle, which has been modified in such a way that it can produce an impulse of its own by some electrochemical processes with the rate of like 72 beats per minute. So this SA node can produce this particular wave 72 times per minute. So this is the power of the SA node, and this is the modification. So priorly it was a muscular tissue, but with time in the embryo it was developed, it was modified into such a beautiful electrochemical kind of unit which can produce its own electrochemical impulses. at a certain rate of 72 per minute. So this SA node is making the impulses at the rate of 72 per minute. And those impulses, they are spread over the heart, especially into the AV node. So this is the AV node. So which is the next part of our, you can say, pacemaking system. Now this yellow spot, it is showing you the presence of the SA node. If I take the second part, which is the AV node, so it is the atrioventricular node, and here we have the green dot, which is showing you the AV node. So this is the second part of the pacemaking system in the myogenic hearts. So if I take the third one, this is the bundle of His. Now this is a great bundle of nerves which are formed again from the modification of the cardiac muscles. And now these are conducting the impulses which are made by the sinoatrial node or the SA node. So we have the third part as the bundle of the His. Now this bundle of His is present at the very junction of the two ventricles and the two auricles. Now it is conducting the impulses which are coming from the AV node to the rest of the heart. So it is conducting all the impulses to the rest of the ventricles. So we have some Purkinje fibers also. So these are the Purkinje fibers which are going up to the whole body wall of the heart. So these Purkinje fibers, they are the seat of, you can say, the impulse conduction. So the impulse is conducted to each and every part of the heart, and wherever the impulse reaches, that part of the heart contracts. Now this is very obvious thing. So if you have the sinoatrial node over here and it is making the impulse, so what do you think? The right auricle, it will contract slightly first than whole of the heart, because the origin of the impulse is in the right auricle. It is by the sinoatrial node. So when the impulse goes from the sinoatrial node to the left auricle, then this left auricle will contract. So as we have studied in the previous classes also, there is a slight gap in the contractions of the two auricles. Now this gap, why there is a gap? Because the sinoatrial node is on the right auricle, and this auricle, it contracts first and then the left auricle contracts. So students, this is a very obvious thing. You should note that wherever the impulse reaches, only that part or only that chamber contracts. Now this is a very, very important thing. Now students, here I must give you some concept about the pacemaking capacity of the pacemaking system. Now what I have is that suppose I take out the SA node out of the heart. Suppose if this is the SA node in the wall of the heart, if I take it out, what will happen? Do you think the heart will contract? So if I take the SA node out, will the heart contract? So many of the students are thinking of, no, the heart cannot contract. But students, the heart has made a provision like this, that if the sinus node is not working, suppose you have some problem in the SA node, and many of the patients, they undergo operation for this particular thing. If the heart is not making a proper impulse, they are not beating at 72 per minute. So what the doctors do, that they take out this particular thing, or they can give a pacemaker to the heart. So they fit a particular pacemaker into this heart, which will be giving an impulse of 72 per minute to the whole of the heart. So that is called as a artificial pacemaker. Now, the God has given us the power of some artificial kind of a thing also. How? Suppose the SA node is not making its beat at 70 per minute. So I am taking it out. Suppose this is an experimental view. This is not a practical view. Now, what will happen? The heart will still make some beats. It will not stop beating. What will happen? The AV node, the second node, will take on the charge of the SA node. So what will happen? AV node has the capability to make a beat at 50 beats per minute. Not at 72, not at 70, but at 50 per minute beat range. So the heart will still beat at 50 per minute rate, not at 70 as the sinus, or the SA node or the sinus node. So if I take out, suppose, the AV node out, if I am taking the sinus node and the AV node out of the heart, what will happen? Still, this heart can beat. How? The bundle of his will make its own beats at about 35 to 40 per minute. So this bundle of his can make still some beats to make the heart contract to some extent. But here there can be some problem in the beating of the auricles, because the bundle of his is in the connection with the ventricles only. So there will be the ventricle contractions, but there will be some problem in the auricular contraction. But I am just giving you the capacity of the pacemaking system. If we have the Purkinje fibers into account, if we take the Purkinje fibers, they can still make their own waves at the rate of 15 to 30 per minute. So that means the SA node, AV node, bundle of his, you can say the branches of his, and the Purkinje fibers, they are all capable of pacemaking. So they have some capability to make the beats. So that is a kind of pacemaking system that is present in our heart. Now students, here this is a kind of, you can say, the beat of the heart, where I have shown the two auricles over here. These are the two ventricles, and this is a beat of the pacemaking system. So we have a, like you can say, a single wave from here to here, which can be written as the ECG, the electrocardiogram. So this is the wave of a single contraction of the heart. Now, if I talk something about the components of the ECG, so these are some of the graphs which are made by the sinus node, the SA node, then we have the atrial node, then we have the AV node, then we have the bundle of His, then we have the Purkinje fibers, then we have the ventricular muscles. That means the whole lot of the ECG wave that we have as a PQRST wave, that is a combination of these waves. That is a whole combination, that is a resultant combination of these kinds of waves. So here what I want to tell you is that there is some component of the SA node that starts the wave. That is a component of the SA node. So then we have the atrial muscles and the AV node coming into account, which are making the P wave for contraction. Now the whole of the contraction, which is the maximum at the R point, it is due to the ventricular muscle contraction. So this part of the wave is due to the ventricular contraction, and this T wave, it is coming because of the relaxation of the ventricular muscle. So if you get a question on the myogenic heart, suppose, So you are not giving this much of thing into that account, but you can give this particular wave only. Now, this is the most important point in the whole heart cycle of a myogenic type of the heart. Now here we take as the right auricle, it contracts first. Now as we have discussed earlier, we have a SA node, then we have the AV node. So what happens that in this particular diagram, you can very well see there are three pathways from which the impulse travels from the SA node to the AV node. Just see, this is the first pathway, this is the second pathway, and this is the third pathway. So from three pathways, the impulse can travel from SA node to AV node. The second pathway, which is coming from the left side, from this particular side, it is giving off a branch into the left auricle or the left atrium. So what has happened that as the length of this particular nerve is quite long as compared to this particular path, it takes some time. So in this time, what happens? The right auricle, it contracts first. So because SA node is present over here, so the wave can spread over this particular auricle very quickly as compared to the left ventricle. So this time gap, it gives a slight first contraction to the right auricle. So this is what I want to say in this particular slide. Now students, we are starting with the physiology of the neurogenic hearts. So this is a very, very important part in the neurogenicity. So before starting this particular part, I must give you some concepts about the physiology of the neurogenic hearts. Now here what we take is these are some of the experiments that are being done with the neurogenic hearts using some chemicals. So as the ACETYL-CHOLINE is you can say a neurotransmitter in our case also, ACH. So this ACETYL-CHOLINE is a very important component in the myogenic heart as well as the neurogenic heart. So what the scientists they are doing that they have given this ACETYL-CHOLINE to both the myogenic and the neurogenic hearts. So they have seen the effect of this particular compound on both the hearts. Now, if a particular heart, say neurogenic heart, it is accelerated or it is deaccelerated by a particular compound, then we can give a particular idea that how this heart is working. And if a heart is accelerated by a particular compound, that means there is some system which is a neurotransmittory kind of system present in that particular animal, which is stimulated by this particular compound. So we have an idea, overall idea, that this heart can work in these kinds of conditions and this heart cannot work in these kinds of conditions. So we have a set of compounds, we can have a set of conditions in which that particular heart will work or it will not work. So these experiments were done to make, you can say, an overall idea that how these hearts they are working. What is the mechanism behind the working of those hearts? What is the chemical nature of the neurotransmitters? What is the chemistry of nerve impulse making? What is the mechanism behind the neurogenicity? What is a kind of ECG that is found in the neurogenic hearts? So we have ECG of the myogenic heart, but what is the type of the graphs which are produced in the case of neurogenic hearts? So all these experiments they have done for exploring these kinds of things for neurogenic hearts. Now students, uh, here we are taking the effect of the acetylcholine. So in this particular lecture, I'll be just talking about the acetylcholine. Now, first of all, as we all know, that the acetylcholine, it is accelerating the myogenic hearts. In our case also, the acetylcholine is a neurotransmitter and it is accelerating our hearts. So if you have more of the acetylcholine dropped into your blood, so that can affect the heartbeat of yours. The acetylcholine, now it has been seen that it inhibits the neurogenic hearts. So this is a, you can say, antagonistic kind of a thing than the myogenic hearts. Now if I go further, the acetylcholine is ineffective at very low concentrations. Now this is a very, very important finding, that if acetylcholine, it inhibits the neurogenic hearts, it should have a particular threshold level of concentration in the blood or in the hemolymph. At very low concentrations, it is ineffective. That means it cannot inhibit the neurogenic hearts, it cannot accelerate the myogenic hearts. Even at the concentration of 10 to minus 5 or 10 to minus 4, it is excitatory. Now just see the, you can say, the power of the acetylcholine. It can accelerate the myogenic heart in this particular concentration. It is highly qualified to be the accelerator of the myogenic hearts. If it can accelerate or excitate the heart at this particular concentration. Now here in this particular graph, what I am trying to show you is that suppose this is a beat graph of a particular heart. Now this wave, a single wave, is representing a single beat. Now the heart is beating like this, beat, another beat, next beat, next beat. And when the acetylcholine is added to the heart, it is given to the heart, what happens? The beat, it is accelerated. So the beat, it accelerates and that excites the heart, and this is an excitatory kind of nature of the acetylcholine in this particular case. Now if I go for this particular animal, this is the example of the Mytilus. It is a bivalve and it is a mollusk. Now here I must give you the idea that the mollusks, they have got myogenic hearts. So if it is a myogenic heart, so ACH is accelerating the myogenic hearts. So whenever the ACH is added to this heart, the beat, it is going fast and this heart is accelerated. So that was the example of the mollusk heart or the Mytilus. So students, in this particular lecture, I am stopping over here about the myogenic and the neurogenic hearts. In the coming slides, we'll be taking up the more of the experiments. So thanks for now.